I hit the button. I made us go live. Blair, where'd you go? Is there a Sadie in the house?
Sadie dropped her treat.
Oh, poor Sadie.
I had to get it for her.
Oh, we always have to be there for our pets and our loved ones.
What am I going to let her get down and pick it up herself? That's ridiculous.
Oh, my goodness. Oh, my goodness. Somebody today was telling me how much they wanted Dogecoin, and I was like, well, I'd rather have a Corgi.
Corgi coin.
Doges. We're live, everyone. This is the This Week in Science podcast broadcast. What?
What are you talking about? I have no idea what the kids are talking about today. You're talking about dogs?
Or am I talking about something else?
Can we pretend cryptocurrency doesn't exist? Just for me, just like
yes,
la.
For the next couple of hours, we're talking about science. We're not talking about those things. All right, we're skipping it and moving in to the show. You ready for the show? Let's do the show. We're going to start the show right now. Oh, yeah. I'm going to hit this button and then that button and then the other button. And then I'm going to start the show in a little bit of 3, 2. This is TWIS. This Week in Science, episode number 823, recorded on Wednesday, May 5th, 2021. Cinco de Mayo Science Salsa. It's very tasty. Hi, everyone. I'm Dr. Kiki, and tonight on the show, we are going to fill your head with regeneration, simulation, and invisibility. But first...
Disclaimer. World governments continue to reflect thoughtfully over the past year's pandemic, waiting patiently to reopen until such a time as it is safe for the general public to return. Like Florida did before leading the nation in COVID cases. Like India did before leading the world in COVID cases. How did the virus overwhelm hospital capacity? Who knows? It was a novel virus. Despite dire warnings from scientists who study such things, it was pretty obvious that it was safe because why bother with facts? Even now, I'm talking about it in the past tense, despite there being more new cases in the world in the past week than any other time, and peak deaths will naturally follow, although actually right before we got to the show, we were at peak deaths globally. So statistically, we are at the worst point in the pandemic's history. But it's not history because it's still happening now. But why bother with facts? Let's just reopen everything. Meanwhile, efforts are underway to reimagine the future of energy production. Our current system was imagined into existence 100 years ago. Since then, we've learned that reliance on fossil fuels, while fantastic at first, is bad for human health. It's bad for land, air, and water quality, turning them toxic, which is about as far from quality as you can get. And, of course, it's also bad for the stability of the climate. We've known all of this for nearly 100 years, thanks to scientists who have been pointing out the dangers all along the way, but still we failed to make any significant changes to our behavior because, again, why bother with facts? Facts are persistent and pesky things, though, and while we may not wish to bother with them, time will come when they don't bother with us either. Because none of the facts we ignore now will matter when we have all sorts of new facts to deal with. All sorts of new questions like, which cities will be abandoned first when sea levels rise above their sewer systems or their suburbs? For what reasons did the superstorms get started? Why are there so many species of animal going extinct so fast? Who knew that minor food web collapse in the ocean could lower the planet's oxygen level? Someday soon, sooner than you think even, the world won't care about warning signs anymore because it will be too late. But it isn't too late. It's not too late yet. There's still time. In fact, you are just in time for another episode of This Week in Science. Coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening.
to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We're back again. Oh. We do this every week, and it's super fun. We get to bring the science.
What? It seems that way, as in it seems it's every week, or it seems like it's fun?
It seems like it's every week, and I think it's fun, but I don't know if it actually is.
I think it is.
Is it? Okay, good.
Yeah, and everyone joining us tonight, thank you for joining us for this episode. I have brought some stories for all of you about viral DNA. I've got some simulated impacts and consciousness. What did you bring, Justin?
Consciousness sounds good. Yeah. I've got invisibility fences, the plurality of posteriors, holographic histopathology, just because it's fun to say, and 3 ways to monetize female fertility today.
Great. All right, Blair, what's in the animal quarter?
Justin brought those multiple posteriors. I know exactly what that story is. I'm very excited to hear about it. I brought Jurassic Garden. I brought regenerating sea squirts, and I also brought teeny tiny stabby shrimp.
Little teeny tiny ones?
Yeah!
I love little teeny tiny stabby things.
Little babies.
Yay! Baby stabby things.
Yeah!
Well, as we jump into stabbing your heart with science on the show tonight, I would like to remind everyone that if you have not yet subscribed, hit that subscribe button wherever you are and get TWIS every single week. We are found on just about every podcast platform out there, and we are on YouTube, Facebook, and Twitch. Our website is twis.org. Okay, it's time for the science. Hot off the steamy presses of the internets. Yeah, I'm just making up all sorts of songs now. Researchers at Flinders University are... Yeah, well, they're looking at plants as their neighbors, and the neighbors that plants live very, very closely with in their new paper that just came out entitled Intracellular Bacteria in Plants, Elucidation of Abundant and Diverse Cytoplasmic Bacteria in Healthy Plant Cells using in vitro cell and callus cultures. What? Wait.
That's abundant? Just the title?
Yes.
That feels longer than the last paper I wrote.
Intracellular bacteria in plants. Okay. It's well-known bacteria can be pathogens to plants. They can get kind of at the borders of cells. They can kill cells. They can take over plants with the microbes of funguses, viruses, bacteria. All these things can get in there, but they don't live happily in the cells in the plants, right? Well, this paper did a bunch of staining and electron microscopy to determine that they were finding abundant and diverse cytoplasmic bacteria. Cytoplasm, that is the body, living in the bodies of the cells. In healthy plants, not diseased, pathogenic plants. We're talking about healthy plants. Play host to all sorts of bacteria. And they found that they had proteobacteria succeeded by Firmicutes, actinobacteria, bacteroidetes, planktomycetes, and 20 other phyla and other candidate phyla. They were able to do genetic tests and RNA tests to be able to determine the species that were present and to contrast them to the actual plant DNA that was present as well. And they don't think this is from contamination. They think that these are bacteria living in the cells of plants, inside the cells.
Okay, many questions.
Yes, so many questions.
Many questions. One, why has this never been? If this is real, this is true. How come nobody's ever noticed before ever? And wouldn't their claim that this isn't cross contamination of some sort from the sequencing? Like, just haven't things been sequenced before? I think so. Like, what is the how? Oh gosh,
what is the how? These are the question. Yeah, so this research has been worked on for over a decade by the involved authors, and this is the authors from Flinders University and also from Australia, and they employed a number of techniques to show that there were basically bacteria around the cytoplasm of cells. However, the bacteria, a lot of times, they're not just all over the cytoplasm. It appears as though they like to hang out near the cellular membrane.
That's where all the action is.
That's where the action is. But so they are more or less like sectioned. They like their bacteria section, but many different methods of showing that they were abundant and diverse. The question now is, can this be replicated? On Twitter, I was informed by a researcher from Canada, Brian Height, that there are a number of insects where it's very common for bacteria to exist in the cells of insects. So we have Buchnera and Wolbachia. They're intracellular endosymbiotes. And there are a lot of symbiotes that are out there. And so finding this in plants as well could potentially open a lot of doors to research in terms of health of plants. How can you get certain plants to grow better? The researchers for this particular study, they're particularly interested in papaya and helping to make the growth and development of papaya stronger, faster, better. Yeah, but it's a whole thing. And then we start asking these questions about these endosymbiotes. At what point do they go away from being separate bacteria, and when do they turn into mitochondria or other cellular organelles? When does that line cross? Yeah.
Yeah. So if this holds, and I have no reason to think it wouldn't, that means we need to look at every kind of, because this isn't in any textbook, the relationship between a bacteria and a plant, in the roots, taking up nutritions, maybe on the leaves. There's a symbiotic relationship there on the outside of the plant. Internal to the plant relationship is not in any textbook, is not a thing that's understood or taken into account for anything plant related, which is a whole bunch of biotech and a whole bunch of textile and food, agriculture, everything industries that take it's huge not to have noticed. That's why I'm also, as much as I have no reason to doubt, also I'm a little suspicious. There's been a lot of looking at this, right? This is not a thing that nobody's
Plants have been under the microscope.
Look at a plant before. Like, that's not a thing.
But see, on the other side, I'm thinking there's bacteria everywhere. It's just everywhere. It's, if you think it's not somewhere, it turns out it's there. Yeah. It's kind of, it's a very cool new kind of thing to see, but it definitely, yes, there is definitely part of me that's like, yeah, of course there's bacteria.
Yeah, which means, of course, step one in looking at plants under electron microscope or in sequencing is you just got to ignore all the bacteria because it's everywhere. It gets into everything. It contaminates everything. So you just ignore it.
So one of the interesting aspects of this study is they were like, okay, we see these bacteria in the cell in the plant cells. Well, let's try and grow the bacteria on their own, and they could not get the bacteria from the plant cells to culture on their own. They only grew in combination with the plant cells, which is another very interesting point, which points to them having some energetic or nutrient requirement that comes from the plants.
So, but then maybe I missed it because I was still getting enough coffee into me to be conscious this morning. Did they identify any of the bacteria as belonging to certain families or something?
Yes.
Like
several specific families? Yeah.
Okay, but have they looked at it close enough to see that it's like close to, but it's different enough because it's been sequestered away for million or 20 million or 50 million years living.
Or not that sequestered. Yeah.
Or just yanked in.
They're in the family. They have the genes that particular family of bacteria have. Genus, yes. And so they're related very closely. It's a species within a genus within.
But these might not be. These were then maybe not captured and then just held captive, and they're up against the cell wall. Like, is there a way out? How do I get out of here?
But then if they get out, they don't survive.
But then they're dead.
What are they doing there?
They must be specifically. Oh, wow.
What are they doing?
This is a great story. Yeah. We need to follow up on it. They need to do more research.
I would like to know more. I want to know more about bacteria living inside of cells, inside of things.
Yeah.
Cellular endosymbionts. Tell me a story, Justin.
Oh, okay. Let's see. Have you ever had wood and thought, gosh, I wish nobody could see my wood right now? Well,
no, I never have.
Okay, well, some people have. Transparent wood was actually developed by researchers at KTH Royal Institute of Technology in Sweden as a new construction material about 5 years ago. Trying to figure out, like, don't we have glass? Why would you need to invent something else see-through? But wood has some characteristics that glass doesn't have. One, it doesn't shatter. It might be more resistant to impact than glass, that sort of thing. But they created wood that could actually let natural light through. It even stored thermal energy to an extent. So the key to making wood into a transparent composite material was to strip out the lignin. I don't know if I'm pronouncing it right. Lignin, which is the major light-absorbing component in the wood. This was first accomplished actually even decades before the Swedish group got involved by a Russian scientist, Siegfried Fink, who was using this technique just to study wood structure. He just wanted to pull it out. But that method left it pretty weak. So you take out all the things, you end up with all these empty pores in the wood. It's nice and see-through. You can sort of see the structure, but then as a material, it becomes very, very weak. So early versions of the Swedish composite, they used plastic polymers that they would sort of saturate back into the wood to fill it. Now the researchers have successfully tested a more eco-friendly alternative, limonene acrylate, which is basically it's a compound that you would find in citrus peels. Basically, they found that they can infill with that; it'll dry clear, and then you can see through wood.
So, what makes it wood and not just a plant-based product that is see-through? Because
that's what wood is, except for the see-through part.
Okay, because straws, plastic cups, things like this are currently being made out of corn, potato, and other plant-based starches.
So no plastic cup has ever been made out of corn. That's the thing. So
there are compostable, plant-based, clear plastic cups.
But then
it's plastic.
How is it plastic still?
It's still plastic. It's a
But I thought plastic was a fossil derived.
No.
Oh.
No, you can have soy derived. You can have.
I guess you can still
potato. You can have corn. Yeah. It just the different type of starch that's used in the for the production is going to have different effects, properties, but. Yeah, so say the decompostable or compostable cups or plates, they're less hardy of a plastic, I guess.
But that kind of goes in line with what Justin's saying about the wood being less hardy also.
Initially. So then they were using plastics, but they wanted to find something more sustainable. So they've used this citrus peel oil compound that they're infusing with now. And they're using this for structural construction grade. So this isn't just, let's see if we can make this see-through, but they're actually doing it to use as building materials in the construction of homes in places where they would like to have light come through. You could have light coming through a wall. You could have light that's coming through ceiling rafters, this sort of thing. And they are at 90% transparency with what they're calling a low haze rate of 30%, which I don't know how you have 90% transparency and 30% haze, but it's a little foggy. But that's still fine. Yeah. Some of the things also, they said there's some pretty interesting and beautiful even light effects that are coming from this. I don't know. It's sort of like you could. Where would you put? Yeah, but where would you use this? My intro there, I was for the naming of the show was calling it like maybe see-through fences. So you want to have your fence so the dog doesn't get out. But maybe if you have all these see-through fences, it sort of looks like much more open space, much more open area in a yard. If you have the
things that you brought up, the idea of having a window that's not quite a window. So instead of instead of a skylight that has glass in it or is made of plastic that can break potentially during a hailstorm or when a tree limb lands on it, you have something that's more structurally sound, and maybe you can have a much larger area of the roof actually is sunroof.
Yeah, and I was thinking about the kind of the haziness of it means that you'd still have some privacy, but you could have passive heating, excuse me, is what I meant to say. So you could, if you had a wall that was made out of this stuff, then you could save a lot of money on heat potentially if it's facing in the right direction.
Yeah, as opposed to having the double glazed windows or triple glazed windows, depending on where you are.
Yeah.
Yeah, you could let that light through, some of that heat through, and some of the versions of this, I've apparently it's very see-through. Now, as you get to greater and greater thicknesses, it becomes more opaque. But some of it is you could read, you could put down on top of a piece of print and read through it just fine. And if you want to check the internet, there's lots of YouTube videos of people doing this at home. I'm pretty sure they're all fake. Except for the ones that failed. There's a bunch that fail that you can be pretty confident they were really trying. But
I think it could be a really great idea depending on it. Right now the cost of wood is increasing in the United States. So wood as a resource, maybe we want this, maybe we don't. But if it is more sustainable and has less of an ecological impact than, say, glass production, if it is better for house heating and cooling, I mean, there are a lot of analyses that potentially still need to be made for cost-benefit analysis. But regardless, instead of a glass brick, you can have transparent wood, which that sounds really fascinating.
Yeah, I don't know that glass is one of those things that they tell you shouldn't even bother recycling because it's so easy to manufacture. And so it's more expensive and it takes more energy to recycle glass than it does to make it or something.
Break
ridiculous.
It and turn it back into sand.
Something like this, yeah. But maybe there's places where this could be really useful. One of the interesting things that I sort of thought up too is maybe there can be an application if it could be clear enough to use it in solar panels. Solar panel glass is already pretty strong, but even if you were reducing some of the capabilities of a solar panel, having a solar panel that has structurally is as strong as a plywood versus a glass might be a benefit in some areas where they're going to be involved in some inclement hailstorms or need something.
All right, Blair, tell us about your story. What do you have coming up next?
Yes. Well, I just rewatched Jurassic Park recently, and it was very fun to rewatch. But obviously, it could never happen for a bunch of reasons. But what might happen is Jurassic Garden. So,
what?
New York University has successfully germinated a 2,000-year-old seed.
Wow.
Yes. So it's a previously extinct date palm variety. It lived over 2,000 years ago, and it was recovered from an archaeological site, this seed. Through radiocarbon dating, they were able to discern that it was from about 4th century BCE to 2nd century CE, so around that 2,000-year mark. They successfully germinated and yielded viable new plants from this extremely old seed found at an archaeological site. So this is the first time that researchers have sequenced genomes of plants from ancient germinated seeds. So not only did they germinate and raise these plants that are extinct, but then they sequenced the genome. So they were able to figure out where they fit and kind of saw how there was some hybridization that happened from the result of the Roman Empire moving into the Mediterranean and bringing plants with them, and all sorts of cool stuff they figured out from the genome.
Wow.
But yeah, they germinated, raised, and then processed the genome for this 2,000-year-old date palm. There was minimal DNA damage. It was a super dry environment. In this case, probably helped it because it was from this dry environment to begin with. But this is the latest breakthrough in quote-unquote resurrection genomics.
Resurrecting a 2,000-year-old plant is no mean feat. I mean, there is an ongoing experiment that people have been talking about for the last couple of weeks. I'm forgetting where exactly it's located, which is probably a good thing because nobody go and try and dig up the remaining seeds that are in this experiment. But over 100 years ago, a researcher decided that we should find out how long seeds can last. And this researcher buried a whole bunch of jars of seeds. And every 20 years, he went and dug them up. And then he died or he finished working and passed it on to another researcher. And so now there's a whole new generation of researchers who are digging these seeds up. And they were able to get seeds to germinate that were over 100 years old from the latest jar that was dug up. But the question is, what are the conditions that are necessary to maintain seeds and their longevity? Like you said, to make sure there's no DNA damage. And
it might be different for different plants depending on where they're from too.
Right. Yeah. So is there something special about this date palm ancestor that made it able, because it maybe started out in a dry environment and it was able to desiccate and it was in a dry environment while it was hidden for 2,000 years from humanity? I don't know.
Yeah. And I love this picture that you shared because it has the kind of the fence around it and a placard, which makes sense because if you're in the middle of the Mediterranean and you just see a date palm, it's nothing special. Maybe you walk up to it and pull a date off or you sit in the shade and eat your lunch. But in this case, it is a very valuable tree. So needs to be clearly labeled and delineated. It's pretty funny.
Very much so. But wow, can you imagine? Yeah, could you imagine a whole bunch of plants like this being brought back in a garden?
Yeah.
It seems like it could be possible. We just need the seeds.
Yeah. Just need the seeds. Go find them, archaeologists. Go get them. I want to see them. Well, in a list of other things that researchers went to find, it was a recent finding in virus DNA that has researchers scratching their heads. Actually, this finding goes back until the late 70s. Researchers discovered one kind of virus called S-2L, an unassuming name to this virus, which infects cyanobacteria. But they found that it incorporated into its DNA a strange amino acid. This amino acid is called diaminopurine, otherwise known as ZDNA. And it is a really strange form of adenine. It's basically the adenine has an extra nitrogen attached to it, and that extra nitrogen makes a stronger hydrogen bond with thymidine, where adenine normally bonds. And so it makes it a much tighter bond in that base pair relationship. Now, they just were like, okay, that's this weird amino acid, and they didn't think they'd find it anywhere else. But lo and behold, some researchers in China decided that they were going to look into this a little bit more, and they have found 60 other species of viruses that use this diaminopurine, this ZDNA, which is an alternative form of DNA, basically. And they're kind of like scratching their heads like, wait a minute, okay, why would you do this? Well, because these viruses infect bacteria, they get into the bacterial cells and they want to cut up the bacterial DNA so that they can insert their virus DNA into the mix or use the bacterial DNA to whatever nefarious purposes the virus has. Bacteria want to defend themselves. So bacteria figured out that they should block certain sequences, certain patterns of DNA from viruses. The viruses that are attacking them, pattern recognition. Nope, you're not going to cut my DNA. I recognize you with your RNA or your adenine, your normal DNA. Well, viruses got tricky, and they decided they were going to be harder to detect. So they made a new form of adenine, diaminopurine. The bacteria don't respond to it. The bacteria don't notice the virus coming in to cut their DNA, and the virus is able to get around their defenses. So this is a chemical adaptation to this predator-prey relationship between viruses and bacteria that has led to an alternative form of DNA here on Earth, in lots of viruses, not even aliens, but here on our own planet. Kind of cool. Yeah. And researchers are kind of thinking about this, and apparently, according to Ars Technica, there is a diaminopurine forms spontaneously in space that was found on a meteorite. So researchers are like, oh, diaminopurine, huh? Maybe that's in the mix for a long time. That's interesting. But additionally, because of the way that the adenine-thymidine base relationship is, the tighter relationship, it's suggested that it might be useful for use in with researchers who are using DNA for computational purposes, for computing, for data storage, or even other purposes where you want to make something that isn't recognizable by cells. Maybe you add ZDNA to the mix to make it get through. Sneaky. The DNA is so sneaky. Sorry, that was my bad French accent making a joke.
No, c'est magnifique.
Magnifique, the DNA. Okay, moving on. Justin from ZDNA, you got some branching things?
Yeah. So first of all, I got to get a quick question. I've been informed by our chat room that Nile Red, YouTuber, has recently done the transparent wood experiment effectively. So apparently this is
apparently it's a thing.
Nile Red is, I think it's a chemist. Anyway, 5 days ago, they did, it looks like he pulled off the transparent wood experiment on the YouTubes. So well done. Looks like he's got a ton of cool experiments up there. So everybody go check out Nile Red on the YouTube. After this show. Don't do it now, because you're already watching this, and then you should check out Nile Red after, because it looks like I misspoke. Also, congratulations, Minion Pam, for a third minion to your own podcast there.
Congratulations! Yay!
Okay, so next story. In the animal kingdom, there are plenty of things worth having more than one of. In Pam's case, she's added another child. Also, teeth are nice to have more than one of. If you're a biter or chewer, more teeth, the better. Eyes, humans have 2, whereas box jellyfish, how many? 24. Yeah, they have 24 eyes. Who knew? Apparently they did. Feet, people have 2. Most land mammals have 4. Most insects have more than that. Very few creatures have more than 1 head, none intentionally. And despite the old saying that 2 heads are better than 1, animal kingdom is content with just really a single noggin situation for most creatures. But what about a plurality of posteriors? What benefit might we be missing by not having more than 1 backside? There is a creature that has more than 1. It's the maritime worm that lives inside sponges. There's one of only 2 known species possessing a branching body. 1 head, multiple posterior ends. This is Ramesillus multicaudata. An international research team led by universities Göttingen and Madrid is the first to describe the internal anatomy of this intriguing animal. Researchers discovered that complex body of the worm spreads extensively into the canals of their host sponges. In addition, they describe the anatomical details and nervous system and reproductive units. It has stolons. Did you read up on this part, Blair?
A little bit, but I'm having difficulty understanding why you'd branch in the bottom half of your body.
And not the top half.
And not the top half, which is the filter feeding side.
Right. You'd want to gather nutrients, not necessarily just get rid of them.
Yeah.
So
this is interesting physiology. Mm-hmm.
There's a couple things with these posteriors. Apparently, that's also where the reproductive organs are.
Mm-hmm.
So
it's part of a budding thing, right? Isn't it part of their...
Well, yes, they create these little stolons, which you normally think of as like a fungus branches out and then starts another little colony off of that branch out. You can have plants that can go and send out these root system that would start off up a new plant. In this version, some of these offshoots then get their own brain and eyes. And then bud off to go be reproductive.
Okay, so this is a way of reproducing, replicating, cloning.
Yeah,
cloning oneself.
I guess.
Yeah, I do believe this qualifies as budding.
I love the idea. It's like, okay, my leg. I'm just going to give my leg its own eyes and let it be its own Kiki. Go off.
Yeah, but posterior first, which is a very interesting way to go.
Yes.
Yeah.
And this is all inside the body wall of a sponge.
Yeah, so which kind of makes sense. If you're going to grow within a sponge, you might need to take some lefts and rights and that sort of thing. But yeah.
Yes. Which, if you consider that it's inside the body wall of a sponge, then it's less like, go, be free, child. It's more like, go 1 centimeter to the left, my child. Still inside the sponge.
Still inside this world, this ecosystem of the sponge. Dream larger.
The marine invertebrate world is a strange and wild place. Stay tuned for more.
I hope this animal is used as inspiration for a sci-fi alien at some point. Doesn't need to be necessarily, but it is interesting. Justin, did you have another story? No, or do I have another story for the beginning? Yes. Branching worms was your thing. Now it's my thing. Okay. So we love this planet we're on, right? And
yes.
we've all seen the has everybody seen the Bruce Willis Armageddon movie where everybody was like, we're going to put a nuclear bomb on the asteroid that's coming to kill everybody and destroy the planet?
I don't think I watched the movie, but it is one of those fantasies that the planet Earth is facing a common enemy, probably, and we all get together and do the rational thing to solve the problem.
In the perfect amount of time, and we are able, it's able to be done, number one, and number two, in the time frame that we have left, we're able to all work together and get it done. Okay, well, NASA JPL were involved in a simulation exercise that took place at the 7th International Academy of Aeronautics Planetary Defense Conference. Planetary Defense Conference. All right, so this exercise involved the discovery of a fictitious, not real, asteroid called 2021 PDC, and it was only used for the purposes of the exercise. Over a period of several days, 4 days, individuals who were involved in this simulation were moved forward through the time frame of what would hypothetically happen in the event of a massive nuclear bomb size or impact strength asteroid hitting somewhere on the planet. So they discovered it, and in the first day, they were able to determine where it was, and they determined that it had a 5% chance of impacting the Earth on or around October 20th, 6 months from the discovery date. On the second day, it fast forwarded them to May 2nd, which was like a week later. And so they knew the orbit and the impact probability, but then they got more information from the Pan-STARRS asteroid survey, found that it had been seen in images several years prior, and that they were able to reduce uncertainties in the orbit and determine its impact. And they determined that from this, the short amount of time before impact, less than 6 months, did not allow a credible space mission to be undertaken given the current state of technology. So they realized, based on, okay, this is going to hit us in 6 months, and they started talking with everybody there, and they tried to run through the simulation of who do we talk to? How do we get a space mission going? Could we, if we could launch something to change its trajectory, could we get there in time, and would that work? The answer is no. And so this is actually a really interesting exercise, because what it means is if we were to find an asteroid similar to this hypothetical asteroid today, no, it would hit the planet somewhere. And if it's in a metro area, it would cause significant damage, and we wouldn't be able to stop it. The only point forward from there would be pinpointing where it's going to land exactly, and then getting people out of the space.
Yeah.
Exactly. Mm-hmm. Yeah. So yeah, they ran through. It was all the top international groups, like basically running through how different planetary defense, space organizations, and individuals could work together in the case of this kind of event. And they worked well together, but it wasn't going to save anybody. So they pushed through it, and they did find in the final days of the hypothetical asteroid impact, they detected it, and they were able to narrow the impact region down to the border of Germany and the Czech Republic and Austria, and were able to talk about disaster response at that point in time. But it's a fascinating exercise to run through, and this isn't the first time that they've done this, but to be able to consider who would be involved globally, what would we do? What could we do? And this is especially interesting since we do have a mission out to an asteroid currently to try to knock it off of its track, off of its orbit around another larger body. And so we will soon actually have experimental evidence as to whether or not the Armageddon scenario is something that's even feasible, because at this point, it's just hypothetical.
So I think if you're going to have them deal with evacuations, you have to also deal with internet rumors that the asteroid is not real and trying to convince people to actually leave.
I'm gonna guess they did not deal with that side
I feel
of
like
it.
This is what we've learned, right? Is we have a global threat. We have this global threat that we're currently dealing with. And there is a whole secondary fight for the global threat, which is convincing people there's actually a threat. So I think in future, that should be part of the conversation too, is how do you, and I'm saying this as if I'm joking, but I'm also serious here that combating misinformation needs to be part of the process.
Yes.
It was absolutely the first thing I thought of when our chat room started discussing how long it would take to evacuate a city. There's 30% that's just going to believe the earth is flat and can't be impacted, right? Like that, this just can't even possibly happen. I think it's a little less of an issue than what we've experienced with COVID, realistically, because part of the push against things like staying at home and distancing and the rest of it is that there are a lot of interests that want frontline workers out there earning money for their interests. And so a lot of the push has been from a corporate side to get the frontline workers out there generating income for them and continuing that money flow. If that money flow isn't there, then I think you see a lot of the politicians who are like, we need the freedom to choose not to wear masks and go out there and let everybody work. I think you see them just saying, whoa, it's going to just blow up the thing. I think everybody get out of there. Then I think people coalesce around the issue better.
Yeah. But I think that the disinformation aspect is something that is a big issue, that whether it's for health, public health messaging, whether it is for politics, whether it is for, getting people to understand that there is a world-destroying asteroid on path to the planet. The disinformation. Yeah, but the disinformation aspect is something that needs to be addressed. And
if it's world destroying, do you even tell anybody?
Don't
even bother.
It's not worth it.
I mean,
Work is canceled for the next 6 months. No reason.
And if it is one of these situations where it's like specifically going to be hitting the Panhandle in Florida, then and nobody wants to leave. It's okay, don't fight it.
What can you do?
Just don't fight it. Just be like, all right, there you go.
All right, everybody.
Florida man.
We are here with information. This is This Week in Science. We hope that you appreciate the information that we bring to you every week. If you enjoy the show, help us spread the word and share it with a friend today. It's time for a very quick and positively focused COVID update.
Yay!
Publishing in the Public Library of Science PLOS One journal, researchers have published their paper called How Well Did Experts and Lay People Forecast the Size of the COVID-19 Pandemic?
Apropos.
How well. So the researchers who were involved in this research project, they saw people asking experts at the beginning of the pandemic last March and April for their forecasting. How long do you think this is going to last? Is this going to be bad? Asking all sorts of questions that epidemiologists and infectious disease researchers, public health officials maybe didn't know the answers to because it was so early in the pandemic and they didn't really have all the information they needed. But because this is an area that they study, they made prognostications anyway. And so these researchers went back and they said, okay, April 2020, let's ask 140 experts in the UK and 2,086 lay people. in the UK. They had to make 4 quantitative predictions about the impact of COVID-19 by the 31st of December, 2020. So how many people were going to die? How many cases did they think there were going to be? They had to give participants had to give lower and higher bounds of certainty of, say, like a 75% chance of their answer being true. How do you think experts versus lay people performed on this task?
Well, the experts probably did better. I would hope.
Whenever it comes to prognosticating the future, I feel like unless you're talking about Vegas odds makers, I don't give anybody better odds on being better at anything. Financial forecasters can't do it. It's the whoever they have doing algorithms for sports betting, those people need to get retrained and refocused on predicting when earthquakes will happen and lightning will strike. Like they're too good.
I'm going to say one thing though before the big reveal here, and that is that I know there are companies that had connections to biotech and experts in this field who, when things shut down in early 2020, they told their staff that they would be there until 2021 at home. Oh. Mm-hmm. So there was a contingent of people that thought that this was going to last this long, and everyone else kind of shoved it off, and people didn't want to talk about it on TV because it was too depressing. Nobody could handle that truth in that moment. But it really did seem like there was a small group of experts that really saw this coming.
Yeah. So in this sample of individuals from the United Kingdom on these 4 questions that they asked, they found that there was a significant difference between the answers that the experts and the lay people actually gave. And what they found in truth is that the experts were better at prognosticating, at telling the future. They didn't get it exactly right every time. And in fact, they weren't close to right, but they were better than all the non-experts.
Wait, did you say they weren't close to right?
No, in some of the cases. So the experts did, in this particular case, the average number of deaths that they expected in the UK by December was 30,000. The actual number was 75,000. Non-experts expected 25,000.
So I'm going to say pretty much no better. That graph that you're holding up there looks like they're running pretty much together.
Right. But there were it was for different questions. They had different outcomes. It was interesting, however, that the experts were much better at the estimation of how many people out of 1,000 were going to be infected, and and non-experts actually way overestimated how many people were going to be infected based on the known infection rate at the time last April. But all this to say, the experts were not perfect, but the experts were much better than the non-experts. Mm-hmm. So even when nobody knows the answers, you're still going to get closer to understanding what's going on when you're listening to the experts. So on Blair's point of disinformation from earlier, when you're talking to people on Twitter who have no background in public health, epidemiology, infectious disease, virology, biology even, maybe you should think twice, and maybe you should try to find your experts, especially when it comes to something like this.
Yeah, okay, that's a good take. My take is we just need better experts. That's
maybe they weren't talking to the right experts.
I think what you need is you need experts and you need social scientists, and you need those two to get together and make predictions, because ultimately I think that's what was missing, is that all these experts were talking about how COVID would work unchecked. And if we do everything wrong, 250,000 people will die in the United States. Mm-hmm. And it was way worse than that. And why was it way worse than that? Because people kept crossing state lines and doing things that were questionable. And I think that if you talk to social scientists, they probably weren't surprised.
Yeah.
So this is where you have to put the quote unquote soft science and the hard science. That's where those things are so closely related and so essential to especially human health, is you have to have this interplay, because those social scientists understand.
When are people just going to be like, I'm not listening?
Yeah. and I bet you social scientists saw that coming.
Yeah. Yeah. I think you are absolutely right. And thinking of the things that we do to mitigate the effects of COVID-19, a new paper is out in Science, Household COVID-19 Risk and In-Person Schooling. So anybody sending their kids to school right now, back to school, there's some risk, right? It's the same. Teachers are going into this situation. People have been working with this risk for some time. Every time you're going into a public situation where there's mixing of more individuals than just the people in your household, you're increasing risk for infection. So these researchers looked at an interesting sample of data which comes from Facebook, which was a survey administered through Facebook in partnership with Carnegie Mellon University. They had about 500,000 survey responses in the United States, and they collected data over 2 time periods, November 24th through December 23rd and January 11th to February 10th. The first is 2020, second is 2021. For the total number of responses, they had about 500,000 survey responses a week. Their total number of responses was 2,142,887 in the 50 states and Washington, D.C. And they reported a number of kids, et cetera, going to school. In-person schooling increased between the 2 time periods, which makes sense. We know a lot of schools did start opening up to in-person instruction in the new year. However, the big takeaway from this study is that there is an increase to the risk of household members. So kids going to in-person school can bring COVID-19 home, and there's an increase in risk to household members from sending kids to school. This study also looked at risk mitigation factors such as mask wearing, as placing desks further apart, using, cleaning methods, all sorts of different things. And they found that each mitigation method that was included reduced the risk to household members. So no mitigation measures whatsoever, you have a pretty reasonable risk to household members from kids being in school. But each layer of protection decreases that risk substantially. And they found that if 7 or more mitigation measures are in use by the schools, there is basically zero risk to people at home. So school administrators, take a look at what you're doing and see how many mitigation measures you can make work in your schools and in your classroom, because the more things you do, the more they add together and protect everybody in the community, not just the people in the classroom.
I still do not understand why we are talking about this in terms of who other than the children might get infected. I don't think we know what a long-term effect is on children who are exposed to COVID. I mean, we act like they're immune. They're not.
Yeah, there are numbers rising in the Midwest of kids getting sick right now too. It's the new variants. Some of the new variants specifically are really bad for children.
And we are learning from the adults who've been infected en masse that there are very long-term effects that come with this. So Basically, they're talking about the vaccine getting opened up for 12 to 15-year-olds soon, right?
Mm-hmm.
Okay, that's when you open the school. Yeah. That's when you send them. Once you've gotten them vaccinated. The idea that we're sending kids out there unvaccinated, like, who's in charge of any of this? There's no one.
It's the same idea. The more non-pharmaceutical measures that you take, there are non-pharmaceutical interventions that can be taken to protect people. And if you are taking a number of these, it's reducing the risk for the kids. It's reducing the risk for everybody.
Yeah, I think that's those mitigation strategies are an important part of the conversation because as you mentioned, one of the biggest factors is how close these kids are together and how air circulation works. Yeah. And if you think about urban school districts, those are two things that you can't really achieve if you're bringing everyone back. You cannot, you have 35 kids in a classroom made for 15, and you have some of them 100-year-old buildings that don't have proper air circulation. So I think this is the importance of this study, right? This is exactly why we need to be having this conversation. Yep. Because if you're just blanket saying, like Justin's mentioning here, kids can't get sick, that's a problem. And if you're blanket saying,
let's make it less.
Oh, we'll do this one thing. We'll put a HEPA filter in the corner and plug it into the wall.
And now we're safe.
Yeah. I think that's what's so important about the study. You said 7 metrics. Yep. So quantifying exactly how many things we have to do to make it safe for the children and their families. That's what we really have to follow. But then again, you have to make sure that those in charge actually listen to this science, which I think is the ultimate hurdle.
Well, which is also why you have to make it simple. And you have to make it so it's one thing that is done to protect the children. And that one thing is get them vaccinated. Before you do that, every conversation about sending the kids back out there is premature.
We should get children vaccinated if the vaccines are proven safe for kids in the age range, because again, it is a risk, and we are working with vaccines that are still not actually approved, but in emergency use authorization. And even though they appear fantastic, the mRNA vaccines especially, everybody wants to make sure that we need to know that the risk to kids from vaccinating is not greater than the risk for COVID. There are lots of factors that need to be taken into consideration.
But we've determined that by sending them back to school. You know what I'm saying? No. I mean, we've chosen one without the information, which is what I'm saying is just reckless. I don't agree with it.
Yeah, well, and ultimately, not to get back on this soapbox, but why is it so important that we get kids back to school? Because we need a place to send children, because adults are being asked to come back to work.
Yeah, they want them to work.
So this is a whole
of the population that wants.
This is a whole cascade related to public health versus economy. That it should not be a versus. It's a whole separate thing. But ultimately, this is good information, knowing that I currently live in a country that is going to put kids back to school next school year. I feel like that's very clear. So having this information will be extremely helpful to be as safe as we can.
Exactly. Yeah, this is the kind of work that needs to be done so that we can move forward even more safely.
And a very frightening thing. Yeah. This is a very frightening thing that took place during this is how many people left the workforce and were on unemployment. And the civilization didn't collapse. In fact, the stock market went up through the roof, and everything's been fine. People have been spending money because they have money, and they're not. Now people have time, and they're doing hobbies and enjoying life to an extent. Like, this has been a great experiment. I'm glad we got to do it. Now they want to end it because it works a little too well.
It works too well. We don't need that. Yeah. Well, that's it for the COVID update for this week. I just thought it was a little bit of good news. Mm-hmm. You know, we've got some information on how to live more safely, more better. But yeah, now I guess I want to say This is This Week in Science. Thank you for joining us tonight. It's wonderful that you are here with us. And if you are loving the show week in and week out, maybe consider heading over to TWIS.org and clicking that Patreon button. Patreon is our support community, and if you are able to give $10 or more per month, then we will thank you by name at the end of the show. There are also fun rewards like t-shirts and other things that we like to send out to you, and we do hope that you just want to hear TWIS every week. You want to see us here every week, just like we want to see you. Thank you for your support. We can't do it without you. All right, everybody, it is time for that wonderful show, the part of the show that is full of the animals. It's Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels, there's Animal Corner.
What you got, Blair?
Oh, I have weird animals from under the sea.
Under the sea.
So the first is a discovery from the Gulf of Eilat. This is researchers from Tel Aviv University discovered a species of ascidian, also known as tunicates or sea squirts, a marine animal commonly found in the Gulf of Eilat that is capable of regenerating all of its organs, even if dissected into 3 pieces.
3 pieces.
3 pieces. And yes, before you ask,
Trisected.
You trisect this little sea squirt, you get 3 sea squirts. So not just one of them is able to completely regrow. You now have 3 identical sea squirts because they can all regrow the 2/3 of them that's missing. So sea squirts are one of the most evolutionarily distant to us from phylum Chordata. So those are animals with a dorsal cord. Basically, it means they have an in and they have an out. Is what makes them kind of different. So there are chordates and there are sea squirts that are able to regenerate organs. There are also ones that are capable of asexual reproduction, kind of akin to the worms that we were talking about earlier in the show. But this particular species that they found in the Gulf of Eilat is one that uses sexual reproduction. So they don't bud, they don't go through fission. They don't do any of this asexual reproduction that might explain why they can regrow 2/3 of their body. These guys go through sexual reproduction. So exactly how they still have these body mechanics is unknown. So they have
they're like, how does it work? We
yes.
don't know.
So let me describe a little bit more about what sea squirts are for those of you that are not aware. Is that they are basically like a living pasta strainer. They suck water in through their body's entry point like a mouth. The strainer filters food particles. They remain in the body for digestion, and the clean water exits through the exit point. They only have one though. And so they have this evolutionary connection to humans because they are chordates, because they have this particular body structure that's like a very, very, very basic human, basically. So, we have an in and we have an out. It's basically the same thing. So they're also known for their regenerative ability. But again, that's mostly been seen in ones that do asexual reproduction. So the first thing that these researchers did is they actually they cut them in half, and they were able to replenish the removed sections without any problem. But they did have 50% of their body. But then they cut them into 3 pieces. So one part of the body had no nerve center. One part of the body had no heart. One part of the body had no digestive system, but they all regrew these complex pieces, and they had 3 identical sea squirts. Wow. So this is the first time they've ever seen, this level of regenerative capacity among a solitary species that reproduces sexually. So the reason I think this is so interesting, aside from the fact that this is kind of the most extreme version of this that we have talked about on the show that I can think of, even beyond that sea slug that regrew its whole body from a head, the tail didn't do that. Just the head.
Yeah.
So this is pretty intense that they can regrow what's missing in 3 different cases at the same time. But it's also important because, as I mentioned, these guys do share a common ancestor with us. So is there a genetic key here? Is there something in common between how they do this and how geckos regrow their tails? Is there something in common between how they do this and axolotls regrow a leg? This is the question that could potentially unlock something very important for us in the future. But the more we can find these similarities amongst distant relatives and their regenerative abilities, the better chance we have of figuring something out in a more complex, let's say, animal.
I'm just wondering when they stopped the experiment. Like, was 3 the upper limit? Or were they?
How many times did they cut the tunicates? Yes.
Yeah. Because would you stop? It's like, wow, you can cut it into 3, and it makes 3 whole. Now let's see if it can do 6. Let's see if it can do 26. Let's see if it can do 126. What's the upper limit of how many pieces you can cut this thing in? It sounds barbaric, but if you're getting all new ones out of it every time, then you can start your own clone army, I guess.
Yeah. So one of the lead researchers says, quote, "Since the dawn of humanity, humans have been fascinated by the ability to regenerate damaged or missing organs. Regeneration is a wonderful ability that we have to a very limited extent, and we would like to understand how it works in order to try and apply it within our own bodies." Yeah. Anyone snorkeling in the Gulf of Eilat can find this intriguing Ascidian who may be able to help us comprehend processes of tissue renewal that can help the human race. So I would guess that, yes, the next steps for this research would be to figure out how small a piece you could cut this thing to still regrow the remainder of the body. Is there a part of this animal that when you cut it so small, it can't regrow? Or is there something that can't be replaced?
What is responsible?
Yeah, which could help us figure out something about the mechanism. But I think also ultimately the mechanism might not be the most important or interesting thing here. I think the genetics and the kind of the key to what triggers this response, I guess, is probably more valuable if you want to think about applications in the future.
Right, because you don't want, if you get a cut on your finger, you don't suddenly want your finger to grow a new finger, right? You have the hand of hands, right?
Yeah. No, I hate.
But it's an interesting question of what kind of damage results in the regeneration of an entirely new body. What kind of damage results in just a limb? What kind, how does that signaling work?
Right.
And I think it's just a really interesting question. How little do you need? How much will it go?
Yeah. And I think that also raises a really important reminder that there are mammals that have regeneration, not of limbs, but for example, bears that can regrow tissue to the point where there's not even scar tissue anymore. So are those similar genetic markers or origins? Is there something there that we can tweak to bring that to a whole other level? Anyway, fascinating, very interesting kind of beginnings. And from sea squirts to mantis shrimp.
Bring it.
Mantis shrimp, baby mantis shrimp, little baby mantis shrimp are still deadly. Yes. So little tiny mantis shrimp larvae undergo 6 or 7 transformations before being seen as what we consider a fully developed adult mantis shrimp. And so researchers from Duke University wanted to take out some high-speed cameras, high-resolution lenses, and, why not travel to Hawaii? Worst places to do research, to investigate the developing mantis shrimp maneuvers. And their research found that mantis shrimp can unleash their deadly blows as little as 9 days after hatching. They look nothing like a mantis shrimp yet, but they can release that killer blow to get their food. So the way they had to test this, I love this. So they had to try to catch mantis shrimp larvae. So sift through a bucket with larval crabs, shrimp, fish, worms, found their mantis shrimp larvae, then super glued them. They're about 4 millimeters in size to a toothpick, then place it on a custom rig to orient them within view of the camera, and then kind of send food their way. It took about a year to figure out how to set up this camera. So that in and of itself is quite a scientific feat. But they figured out how to set it up and rig it just right so that they could collect this data. And what they found was that they actually could see the region on the first portion of their appendage where it bent to store energy like a spring. And then as the larva wound in the club-like limb, it would flick it out. It would release an internal latch that held the appendage in place, released that stored energy, catapulted the limb into action, and it looked very similar to how adult mantis shrimps use this is the club-like action. And so they could see tiny muscles in the larva's glassy bodies contracting as they bowed the exoskeleton. And this is really special because you can't see that in adult mantis shrimps. So they set out to figure out when this develops when they're growing up. And instead they learned more about the mechanics of a mantis shrimp club just overall, which is a super cool accidental find.
They discovered that it's there all along.
Yeah, and they could see it because there was no thick, opaque exoskeleton in the way. And so these 9-day-old hatchlings, their limbs reached a acceleration of 22 million degrees per second squared, and it's about 0.385 millimeters a second. So that's actually 5 to 10 times faster than the little larva that they were eating, and it's very close to the adults. So they had about the rotational speeds were about 16,500 degrees per second. So all this to say, the limbs moved slower than the adults, but it was still similar rotational speeds. It was just slower because they're tinier. So the actual acceleration versus the rotational speed. So there's a whole physics. But anyway, similar mechanics, similar acceleration, different rotational speed. So slower than the adults, but still extremely fast, which means that they have no problems getting their food.
The quick little ones, they're already punchy.
Mm-hmm. Yeah, and so so I didn't even mention. So how did they figure out exactly when this developed in the mantis shrimp? So they found a female who had eggs. They retrieved her. They took her eggs. Sorry, ma'am. By the time they arrived at Duke, the eggs had already hatched, but they were able to nurture the little babies. They were able to develop to 28-day-old larvae, and they were able to kind of test their reaction time to feeding throughout all 28 days. And in the 9 to 15-day mark, they were able to manage these intense reactions to food. So they confirmed they found it just by grabbing larvae in the wild. And they were like, all right, when exactly does this happen? And it does happen very, very fast. Mantis shrimp's got to eat.
A lot.
Mantis shrimp's got to smash.
Got to smash and eat.
Mm-hmm.
Smash to snack.
Yep.
Yeah. Thanks, Blair. That's interesting. That's
there's
dangerous.
David Attenborough special on the Netflix right now talking about colors in the animal kingdom.
Yes.
And there's a peacock mantis
shrimp
in
has
there.
12 different kinds of cones.
But that takes on a crab, and you get to see a strike and what they're capable of, and it's scary.
Mm-hmm. It's
small. I don't feel threatened.
If you have a very I don't know, not sick sense of humor, but if you want to see people get hurt, you can watch people who've decided to take in mantis shrimps as pets, which it's a terrible idea, which is why it's kind of like, okay, they kind of deserve it. But these people get mantis shrimp as pets, and then they think it's fun to try to hand feed them. And they get stabbed or smacked, and often it also breaks the aquarium.
Oh my goodness.
So it's a good reminder of why that is not a good pet.
Okay.
Is when I worked at the zoo, we would have good pet, bad pet conversations, and I would say mantis shrimp definitely
not a good pet. No.
Just leave it to the researchers to have the mantis shrimps in the aquarium.
Please do that. Oh, and you out there. You listening to the show. Not you, but you. All of you. You. Sitting there, listening.
Feels like you're talking to me.
Yes, you. If you enjoy TWIS, why don't you tell the world with a little bit of TWIS merchandise? Head over to twis.org, click on our Zazzle Store link. We've got some new items in the store, right? New mug and t-shirt. Full of science-y goodness. Go check that out. We got science-y goodness in our store. And now, Justin, it's your turn. Tell me a story.
Let's see. I don't know which is the one that I had. Oh, I think I know. Holographic histopathology. Holographic histopathology. This is a story I selected just so I could say holographic histopathology.
Is that Beastie Boys that you're doing?
Yeah, a little bit. Histology, for those of you who are not familiar, is not the study of drunk history episodes, but the study of biological tissues at the microscopic level. The science behind that magical, mythical lab where the biopsies are all sent off to is histology. Tissue samples are collected from a patient. They are examined for signs of disease using a process that requires this tissue sample to be stabilized, sliced, stained, fixed to a slide so that the anatomy of the cells can be clearly visible under a microscope. The process itself, turnaround time between when a sample was taken and is actually been analyzed can be days between. Now, a high-resolution imaging technique is hoping to speed up and improve the turnaround of the sample processing with the power of optical diffraction tomography. Research team has optical diffraction tomography, which, if you're not familiar, is a microscopy technique where complex scattered field light is transmitted through the sample, then retrieved using off-axis holographic scattered field illuminations mapped to a Fourier space on the back of a reflective index, then folded into the shape of a crane. Basically,
optical origami.
Optical origami is sort of, let's just call it that, and it's easier. Basically, they managed to get a really good 3D image of 100 micron thick tissue sample, which is compared to normally the slides upon which they're operating to do the sampling, to do the observations, is about 5 microns. It's very thin slices that they make to create sort of a 2D slide of the tissue samples that they want to look over for pathologies. This allowed them to use 100 micron thick tissue sample and get a little bit of a 3D imaging in there by the way that they were able to do this. And And in some of the cases, they could do this anywhere from 40 minutes to a little over an hour and a half. So they also had a pretty quick turnaround on this. There's still issues and stuff. They still need to work everything out. But one of the sort of neat things that this would allow, a lot of times the reason that you're going to have these biopsies and the like looked at is to see if you have a disease, or to verify that this tissue looks diseased. Let's send it in to confirm it in the lab setting. But the other thing that can be used for, if you have very fast turnaround that has very clear looks, is if you have had then a surgery to remove a tumor or something of this nature. You also then want to make sure you've removed all of it. So you might take a biopsy of surrounding tissues, and you want to have a quick turnaround where you can see this right away. You still got the patient there. You still got a way to get back in and attack this if you've missed. But if you can get in there and look at tissue samples in almost real time, very near real time, to get a good view of it, you can also then sort of make more efficient surgeries when you're trying to remove diseased tissues. But pretty big advance there once it's operational, and I'll have some real benefit in saving lives. But mostly it at present is just allowing us an excuse to say holographic histopathology. Holographic histopathology.
I like it. I, having taken histology labs in school, the idea of being able to look at structures in tissue in more of a 3D image, that is thrilling to be able to see how some of these ducts and vessels, how they look, not as flat, because flat is not what we are, but we that's what we tend to see.
Are three-dimensional creatures.
Yeah, fascinating. Oh, that's great.
Don't let the screen fool you. We are three-dimensional in our nature.
We are in our real lives. You had one more story?
Oh, yeah. I was going to give tips on how to monetize female fertility.
Why?
So, well, because everybody's got to make a buck these days. It's the new digital economy. And so apps for women in which they track their fertility are being utilized as marketing tools. This is Newcastle University and UMA University. I found that the most popular top fertility apps have been collecting and sharing data without user knowledge, permission, or consent.
Hundreds.
Yeah, not good. Not good. There are ones out there that they're probably better than others. There's some that make very extensive, detailed claims about their sharing and not sharing and all the rest of this. But what this research found is at least some of the top ones that they found through the I think they Googled, like the top Google recommendations for these weren't even we're sharing, what was it, 3.8 tracking trackers were implemented and operating before anybody had even seen a privacy document.
Wow.
Yeah. This is Dr. Miriam Mezuzad, Newcastle University School of Computing, Dr. Teresa Almeida from the Department of Informatics at Umeå University in Sweden. They looked at all these privacy risks, and they saw a lot of misuse and misappropriation of what they're classifying as intimate and basically health records is how they would classify this. So one of the things they also noticed was that there are rules and regulations that are supposed to be present in this digital environment where you have to get permissions to use things. They found that those in these top apps were being completely ignored, just completely ignored.
Why am I not surprised?
Yeah, the study showed that the majority of the fertility apps were classified as health and fitness, some as medical, one as communication. The authors argue that miscategorizing an unsecured app which contains medical records as health and fitness would enable developers to avoid potential consequences. Data could continue to be sold to third parties for various unauthorized uses, such as advertising and app development. They're also concerned about other ethical potentials of having this data exposed to people that the women may not want to have it exposed to, especially when you're talking about they're putting everything from temperature to emotional to sexual experience, activity, orgasm, mood, pregnancy, terminated pregnancy. All of these things are ending up in this these apps that help people track. If you're trying to get pregnant, if you're trying not to get pregnant, if you just want to sort of compare your sexual activity, health responses from one month to the next,
or maybe you're just tracking. Maybe you're just tracking your cycles for.
I have to tell you, I'm a woman and I use one of these apps, and it's extremely helpful because if I'm having a day where I'm like, I hate everyone, I think everyone hates me, and also I feel tired and everything hurts, I can open the app and be like, oh, that's why.
That makes sense.
Yeah.
and then also your significant other can also like have a very, so have you checked your app lately? What app? What are you talking about?
He wouldn't dare.
I can't say any more than that. But I just maybe check the app.
Yeah, I mean, I think it's maybe if you're going to be using an app like this to track yourself, I mean, not even just look at the permissions that you're giving and make sure that they're not going to be taking your data and using it for marketing.
I have to tell you, I feel like
an app that isn't going to do that.
I feel like anonymized data that would be actually really nice to have. It would be cool to have an app and to hear people say, that this time in this cycle at this age, 60% of women go through this thing. Right. Or for people that are on our app, it has taken people on average 18 months to get pregnant. This would be helpful information to have if you think about that kind of use. But to use that data for selling stuff or to target with other things, it just, I think that's the part that gets kind of
the commodification. Yeah, it's every app is there to make the makers of the app money, and beyond just potentially paying for the app, there's they realize that they have this data and will potentially use it. I think it would be amazing to have a marketplace of apps that didn't collect all the data so that it could be taken and used for marketing, and maybe you just pay for the app and you can be a part of crowdsourcing information about different life phases and medical histories and anonymize the data. But everything is commodified, and that is the hard part.
We are coming upon an age with the next iOS, the operating system from Apple supposedly is going to have opt-in as opposed to opt-out of a lot of this stuff, which would be a gigantic improvement.
Yeah.
But again, in this case, when they were looking, there wasn't an opt-in and an opt-out and inform. It was just you signed up for the app. So now it was an average of 3.8 trackers per app started right away. So then it's going to be your Google suggestions, your Amazon purchase banner ad things, all that kind of stuff is going to start populating. And it's...
Yeah, and I think this makes me sad because, I'm seeing a lot of side chatter in the chat talking about whether this sort of thing is really necessary. But I will tell you that it has helped me understand my own bodily processes and cycles in a way that a calendar wouldn't have done for me. That an X on a calendar would not have been the same. That an app that follows you for, if I have had this app for probably close to a decade, it knows how my cycles work, what their average length is, when certain things happen. And it has helped me so much to be able to recognize what is going on inside of my own body in a way that we didn't really encourage people who have menstrual cycles to look at closely, potentially in the past. So I think there's something really beautiful to learn through this.
Blair, just keep an eye on the ads that are served to you.
Yes.
On various platforms.
Mm-hmm. Yeah.
They know.
Absolutely. Yeah. And I think that's really frustrating because it's something that, mine is created by Planned Parenthood. I kind of assumed that if it was created by Planned Parenthood, that it would be a pretty safe app to have.
It might
but
be.
Guess what? I'm going to go in and I'm going to look now because that was my assumption, but I don't know. I should double check.
Yeah.
Mm-hmm. What data are they collecting? That's it. Yeah. We should know these things. Oh, everything's a commodity if you look at it the right way. But not bats. Bats are just awesome and batty. 2 stories were out this week on the biology of bats, the physiology of bats. One study, the first, wanted to find out whether or not bats have an innate sense of the speed of sound, or whether they're born understanding how the sounds they make bounce off of objects and can understand that innately, or whether they have to learn it. And so how do you test something like this? Well, of course you raise bats in an environment that has helium in it.
Huh?
That's right. So researchers with Tel Aviv's University School of Zoology, they were looking at the echolocation in bats, and they reared bats, some in a normal enclosure and others that had helium added to the air. Helium's thinner than normal air, so sound goes through the air faster. And they were able to test the bats when they echolocated on objects a certain distance from them to see what their responses were like. And they found that the ones that were raised in and flew in the helium-enriched air underestimated the distance to the target. They were not ready for the effect of helium. They just weren't ready for it. And so they did a second experiment where they went and they caught wild bats and brought them back to the lab and then did the same test with and without helium in the air. And again, the bats underestimated the speed of the sound. So if they had been trying to catch a moth, they would have totally missed it. They wouldn't have been anywhere close. And what it suggests is that bats are born with their ability to echolocate and know the speed of sound. That it's in their ears. They're calibrated from birth. Yeah. Yes. Which I thought was super cool. And then the other study looked at the magnetic sense of bats and whether they're looking at this interesting idea of orientation and how animals orient at night, especially animals that migrate and maybe at night are traveling by the light of the stars. The research published in Communications Biology from researchers from Leibniz IZW, they demonstrated for the first time that environmental signals are important for navigating over long distances and that these signals are picked up by the cornea. And in this case, they used Pipistrellus nathusius bats while they were migrating, and they anesthetized the cornea of the bat's eyes. So they basically put the cornea of the eyes to sleep. They know this is safe because this is the same kind of anesthetization that occurs in eye surgeries by ophthalmologists. It desensitizes the cornea. So it's you get anesthetized and your cornea isn't going to be able to adjust to let light in or out. It just relaxes. And our cornea is very important for monitoring and managing the amount of light that is let into the eye to be focused on the back of the retina. In this experiment, they used lidocaine to anesthetize one eye or both eyes, and then they had captured these bats in this migration corridor in the Baltic Sea, and then they took them inland and released them in an open field. They had volunteers who released the bats and had no idea which eye or which eyes had been anesthetized, but then they had to release the bats into the sky and see which directions they went, with the assumption that if the cornea was involved in navigation, that those that had their corneas anesthetized would not migrate, would not move in the right directions. They wouldn't be able to orient according to the light, the stars in the sky. And what they found is that, yes, indeed, the corneas were involved, but if the bat has at least one eye working, they'll be able to orient just fine. So it's with losing both of the corneas, made it so that the bats could not navigate. And they kind of went all over the place until the lidocaine wore off, and then they flew in the right direction. So what they say is this evidence in different behavior suggests that corneal anesthesia disrupted a sense of direction, yet orientation apparently still works well with one eye. We observed here for the first time in an experiment how a migrating mammal was literally blown off course, a milestone in behavioral and sensory biology that allows us to study the biological navigation system in a more targeted way.
Okay, well, that's pretty cool, except is it really like that stunning of a breakthrough that if you blind something, it has a hard time navigating?
Well, it's a common misconception that bats can't see.
Yeah.
They can see and they use light very well. And the second part of the experiment, they actually, to test that it wasn't just light that was the issue, they have a Y maze where one end has light in it and the other doesn't, and the bats show a preference for light at the end of the tunnel. And so when they were released into this Y choice maze, it didn't matter which eye or any eyes, the bats all chose the light at the end of the tunnel. So the anesthetization didn't affect their ability to detect light, but it affected their ability to orient. And so it's this orientation factor, not being able to go in the right direction, that the researchers are now saying it signifies that this is the bat's magnetic sense, that the magnetic sense of these migrating animals is in their cornea.
Yeah, I still think that's a stretch. I think look, if your eyes are dilated to where you can see a bright light while walking, you can sort of stumble your way in the right direction with your arms out, making sure you don't bump it. Now you're gonna fly. Now, okay. This is a whole extra
fly
level.
All over.
Yeah, this is. I mean, I would like them to have done this right, and that this is the result, and that we've figured this out. But I don't know.
Well, it may not be magnetic sense, but why? But what is so? Orientation is affected. One eye not affected. Both eyes very affected. But it didn't affect their ability to see light, to actually detect light.
Well, but that's such a low bar.
But how much light? This is again, like you say, like this is the lidocaine in the eye. If you've gone through an eye exam and had your eyes dilated, what exactly does that do to your nighttime vision, right? And how would that affect your orientation, and why would it affect your orientation? And this is an important question here, I think.
Yeah, I think if they're looking for the site of any sort of magneto perception, the eyes just seem like evolutionarily, right? It should be in the inner ear. If it's anywhere,
you would think
it should be in the inner ear.
But haven't we had a number of studies now with pigeons and others where it's thought that there's the magnetic particles or other things that are near the beak or in the eyes themselves that create something of a glow that could be perceived through the neurons in the eyes as added light to certain portions of the visual field, where if you are lined up, I say you as if you are the animal. Yes. If you are lined up with magnetic north, that perhaps you have more light, more aglow in that portion of your field when you're looking. So this is why the eyes are something that is considered. But yeah, is it the cornea? Is it? We still don't know. But this is a very interesting mammal-based experiment that again indicates that a certain part of the eyes is involved in orientation. Perhaps. Maybe it still is just an artifact of the fact that they were dilating these bats' eyes.
Yeah, it feels like that. So but you know, like, has there been just like a really basic experiment where they have a treat? They have like a couple places where treats can be put, and the place where the treat is always has a really strong magnet. One of those magnets you got to be careful. You can't play with, because if it gets on your refrigerator, you'll never get it off. Kind of a thing. Like one of those really strong
neodymium,
magnets.
yes.
Yeah, but whenever that magnet is in one of these containers, there's a treat there, and it's every time. You just train them on that a couple times, and then put those treats out there and see if they always fly to the one with the magnet. Bats, pigeons, whatever it is. Have we even started with the basics? Like, how do we know?
Yes, that's interesting. I think it's less about attraction. It's less about attraction to magnets, and it's more about sensing a magnetic field for orientation.
Yeah.
Right, but this is my point. Like, if you created a treat reason for a specific field, then maybe. But usually, like, I'm not. There's even been.
They have disrupted magnetic fields you can set up chambers where you have a strong magnet that overpowers true magnetic north, and they have been able to shift birds and other animals to get them to fly the wrong direction because of shifting these magnetic fields. And so there have been experiments that have definitively shown that magnetoreception is a thing. We just don't know the mechanism. We haven't figured out, and it could be different in different animals for all that we know. It could have evolved differently in different species, but it definitely is a thing. But how does it work? Magnets, how do they work?
Magic.
Yeah. Especially when bats are involved. But I thought those two studies were pretty fun. Bat biology.
But in the same one, you could dilate the bat and have the magnetic treat at one end and the non-magnetic, and then they could crawl to it. They wouldn't even need to fly. So you could tell whether or not they needed their vision to see the magnetic. Just like there's other ways to do the design in the experiment. I don't think you need
to throw the bat
make something
into the
blind
sky,
and throw it into the don't make them fly blind. That's not
blind
fair.
The bat and throw it into the sky.
Oh, that's so wrong.
My final story for the night is all about consciousness.
Oh.
Consciousness. What is it? Well, it's the technical description of consciousness is that it is your awareness of sensory input and behavioral action. So your brain, what is going on when you're conscious. You don't know what is going on when you are unconscious, right? Well, researchers just published in Cell Reports their study looking for the part of the brain that allows consciousness to happen. Basically, the gating mechanism, the keymaster, the gatekeeper for consciousness in our brains. And they used propofol, which is an anesthetic to put volunteers into various states of unconsciousness and ask them to do various neural exercises, mental exercises of imagining animals, imagining navigating, and then imagining squeezing their hand, and then actually squeezing their hand. And so they measured all this in functional magnetic resonance imaging to be able to get an idea of what areas of the brain were active during certain actions and in changing from one action to another, from imaginary actions to physical actions. And in the process of this, what they eventually had people do was they had them look at a screen in which they were going to be shown just like some fuzzy graphics. But right before those fuzzy graphics started, there was a quick picture of the face. So fast, it's just barely visible long enough for a brain to pick it up. And while they were flashing that face on the screen and then having them look at pictures after that, they later asked the people if they saw the face, when they did and when they didn't. And so this is the conscious recall of, I saw a face, or what face? I didn't see a face. Anyway, the researchers then found, by looking at their functional magnetic resonance images of the brain, that there was one particular part of the brain that stood out among all the others. And it's a little teeny tiny part of the brain called the anterior insula. And the anterior insula has been implicated in all sorts of things like emotional regulation and concentration and a lot of things. It has connections to our attention network and our default mode network, which are important in the switching between consciousness and unconsciousness. People have thought about it as a possible contender for the seat of consciousness previously, but in this experiment in which they played with people's conscious awareness with propofol, making them anesthetized or not anesthetized, and playing this imagery to them versus having them be conscious of seeing a face or not seeing a face, it was this anterior insula that popped up. And they are pretty positive that this is the part of the brain that if it's active, it is shunting information from your sensory system up to the higher levels of your cortex for processing and awareness. And when it is turned off, you aren't going to know anything. You're not conscious. Yeah. So we found it. The seat of consciousness. It's in your brain. And it's not like a whole, consciousness isn't, it is your whole brain working together, but there's a part of the brain that's important for what you know and what you don't know.
Your anterior insula.
Your anterior insula. A little tiny thing.
Is there something like this in animals?
And I think that's the next big question is which animals
great question.
would have a structure like this. Yeah, so that I think is the next big question. And I'm sure that comparative neuroanatomists would know the answer to this question already. Mm-hmm. Yeah. Actually, let's stop sharing that. I know it's late. Anterior insula.
Neurobiologist is on the case.
Yeah, I don't know. So this is it's an interesting question. I'd love to find out if it is something that there's an analogous structure or something similar in other species.
And is just having the structure enough for us to say that the animals can do the same thing, or does it have to light up in a certain way at a certain time? There's all sorts of... Yeah.
Right. And even if they have this structure, is it exactly the same? Yeah.
And they might have consciousness, and it might be in a different place in their brain because it might have developed convergently.
Right. Anyway, it's a very interesting question. But now this is the part of the brain that, if you're taking a mental break, is going to sleep. When you go to sleep tonight and are not conscious for a long time, it's not just your pons and your lower brain that's turning everything off. It's your anterior insula is probably taking a big break as well.
I'm sorry, can you repeat that? My anterior insula.
Oh no, it's already happening.
My anterior insula says, it's time for us to end the show.
Woo!
Woo. But before we go, I do want to say, those of you who are on the eastern seaboard, you're in for a big treat in the next week or so. Brood X is coming.
Treat. The cacophony.
The cacophony of the cicadas is coming. There will be billions of them in the eastern United States. This is the population of cicadas that take 17 years to emerge. So these buggers have been underground for a very long time. They're going to come to life and do all their mating excitingness and then be a lot of food for animals for a little while. And then they're going to go back into the ground, into their burrows. Yes. And the eggs will be there. They'll hatch and turn into nymphs, and 17 years from now it'll happen again.
Oh, it's 17
exciting.
Years already. Can you snooze it for another year?
Yeah. One more. Nope, this year. No snooze button. This year. So I'd love to know if you see cicadas emerging wherever you are. Brood X is on its way.
100 decibels, louder than a leaf blower or a lawnmower.
That's a lot.
God, cicadas are so loud. The first time I heard them, it was unreal.
Yeah.
I had to ask somebody what it was.
What is that noise?
Oh, jeez. Oh, yeah. Yikes.
Well, this is the sound of This Week in Science coming to an end. And I do want to thank everybody for joining us for another episode of This Week in Science. And I want to take some time to thank the people who helped this show out. Thank you to my co-hosts. Thank you to Fada for doing social media and show notes. Thank you to Identity 4 for recording the show. Thank you, Gordon McLeod, for manning the chat rooms. Rachel, thank you for your assistance. And thank you to all of our Patreon sponsors. Thank you, John Ratnaswamy, Kira, Carl Kornfeld, Melanie Stegman, DeKramsta, Karen Tawzy, Woody MS, Andre Bissett, Chris Wozniak, Dave Bunn, Vegard Chefstad, Hal Snyder, Don Stylos, a.k.a. Don Stylo, John Scioli, Guillaume, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Shubru, Darwin Hannon, Donald Mundis, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredis 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Kevin Railsback, Flying Out, Christopher Dreyer, Mark Mazzaros, Artyom, Greg Briggs, John Atwood. The profile name is hilarious in the context of some other podcast. Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, and Matt Sutter. Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Sarah Chavez, Sue Oster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Pecoraro, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts.
That is a longer list, and I love it.
I love it. Thank you, patrons. Thank you for supporting us. And if you are interested in supporting us, you can head over to twis.org and click on the Patreon button. In the meantime, we'll be back next Wednesday.
Yes, we will be broadcasting at 8 p.m. Pacific time 5 a.m. Central European Time from our YouTube and Facebook channels, as well as twist.org/live.
Hey, do you want to listen to us as a podcast? Just search for This Week in Science or where podcasts are found. And if you enjoyed the show, hey, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twist.org. And you can also sign up for a newsletter if you're so inclined.
Yes, one coming out soon. You can also contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put twist, TWIS, in the subject line, or your email will be chopped into 3 equal parts. They will grow into 3 separate emails, and we will not read any of them.
Oh, no. If this happens to you, please try to reach out to us on the Twitter, where we are at Twist Science, at Dr. Kiki, at Jackson Fly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views. But I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought. And I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
So, how do I put things in the store that I made? Why is Zazzle so complicated?
Let me look. Let me log in and see what's happening.
Why is not where I want it to be? I wanted to... Zazzle. It says they've been posted for sale, but they're not in my store.
Okay, let me look. Zazzle.com/This Week in Science. Okay. How did you make them is my first question.
Yeah, I don't know.
Did you just start with a blank piece, or did you like copy something that we already had?
No, it was a blank piece.
Okay. So let me see, because you might have made it for yourself and not to sell.
Well, I got an email that says, oh, maybe it hasn't been, I did it yesterday. It says it should appear in the Zazzle marketplace within 24 hours.
Oh, okay, so it needed to be approved then.
I don't know.
See, is it under review? Reviews.
It's confusing.
No, that's not what I want. I want
Here, I can...
my order. Oh, they changed it.
Yeah, it's too...
Oh, they changed the interface. How funny.
Yeah.
Sell. Let me go. My stores. This store. This is what I want. Okay. And then products. And then... Hi, Sadie.
Hi, Sadie.
She's watching her father leave.
Here, I can show you...
Yeah, show me. Oh, yeah, this... I forgot I made the stretch pants. Oh, my God, yes.
Late night stretch pants. Those are fun. Here. Yeah, I made... It's just a cute... But I made a t-shirt also.
Oh, full of sciencey goodness.
Isn't that fun? Full of sciencey goodness.
It's not in the store yet?
No.
Okay, so let me see. So products.
And I also have a t-shirt.
Where is it's? I've looked at the under review before, and also let me say I have had things that had never left review, and I've sent multiple emails about it.
I don't even think it's in under review. I don't know what's going on. Zazzle confuses me. Confuses me. Don't you want to be full of sciencey goodness?
Collections.
It's got the TWIS logo on one side, and then you can be full of sciencey goodness with a donut on the other.
Oh, continue. I don't think it's fully posted yet. Okay.
I got an email that says it is.
Sell it.
So confused.
All options, events and occasions, recipients, store category, new products, tags, suitable audience, show customize it button, royalty %. I have the right post.
Anyway, there will be something showing up soon.
I think it might be in there now. Let's see.
Did you find it? I posted I bet I posted it to Twitter.
Oh yeah, it's all in your head hat. I have tried to sell multiple times, and it's so they're in Save like, Designs, no. And they're like, no. That
is my T-shirt in Save Designs.
Here I'm looking.
No, here.
Under review, I found it. No, there's nothing. The only things under review are postage stamps, which they pulled at some point, and I think it's because they stopped working with stamps.com. Yeah. And the embroidered polo because they stopped selling the polo that we had it on.
Yeah.
So now, where's my store? My store.
Let me see. I found it. I've got it, but I don't know.
Oh yeah.
Got the T-shirt.
Oh, there's no sell it button. That's why.
Why?
I don't know.
Oh, we need one of those.
Where
it says designed for you by This Week in Science right?
Designed for you to buy. Is it like a one-off purchase that you're setting up by accident?
Maybe I just don't know how to use Zazzle.
Yeah, so the mug is now in the store. Awesome. So how'd you get to that T-shirt? Where was it?
This is a link that Zazzle sent me.
Oh, weird.
And said that this is what's going to go in your store. Okay. I don't understand.
I see you.
Blair Baz is the Zazzle master. just let Blair do it. Do all that stuff. But they've got it set up so there's like this stuff on the side now, and then they've got.
They totally changed the interface.
They've got this thing.
My stuff.
Very cool. I like it.
Right, and then in the back has the twist thing.
I need that one.
Full of science and goodness.
Okay, I gotta go.
Okay.
Where are you going?
I'll see you guys next week. Breakfast and then bed.
Good planning.
That's the order. Show, then breakfast, then bed.
Perfect. Seems a little backwards, but that's just me.
It's just this week.
All in a good day. All in the good day.
See you guys next week.
Viking.
Okay.
See you, Justin. Have some good rest. I hope you sleep better during the day.
Yeah, maybe.
Maybe. Who knows? Take care.
Where is your T-shirt? I don't know where it ended up.
I don't know.
Save design.
I had an email. I don't know. These things, it's not making any sense to me whatsoever.
Under review, revise and resubmit.
Yeah. Maybe that's what you...
I don't know where that T-shirt is.
Right? I don't know where it is.
That's really weird.
I just forwarded
Oh, you forwarded it to me?
The Zazzle email to you, so you can at least have the direct links now to play with.
I have to put some
I should never
some of the newer images
to do anything.
No. That's no. You forwarded it to me? I didn't get it. Oh, is it? Nope, didn't get it.
Oh, I see why. Never mind.
Did you send it to the wrong email?
I sent it from the wrong email.
Oh.
La 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 la. There we go. Ta-da.
Yeah, Noodles,
Super
it's
fun.
not, it wouldn't be the supply. If it was a supply issue, it would still show up in the
No, it's just like a weird
account.
system issue where it's not showing up, and I don't understand why. I was very excited about these new shirts, though. I think everybody should have a full of
Here we go.
sciencey goodness shirt.
Your designs are now for sale, it says.
That's what I said. Yeah, that's what I thought.
Okay. Designed for you by This Week in Science. Full of sciencey goodness. View all products. Yes.
Oh, no. Oh, that was... She changed her name again. Nadelva is the accidental Viking.
Oh.
Carol Ann, right?
Oh, yeah.
Do I think AAAS 2022 will happen? I hope so. I can be optimistic and hope. I think a lot, they will want to really be planning it right now. So it's just hard to figure it out. I think it really all depends what happens in the fall.
Yeah.
I feel like it's kind of the summer. We have to kind of wait and see. And then when everybody goes back to school and goes back to work and the winter starts to come again, I feel like we have to kind of see what's going to happen. I think a lot of live conferences and live events need to figure that out. I mean, there's also not just having the live event, but also determining whether or not you want to be a part of having people travel across the country and, and how do you do that? I mean, we don't have the, vaccine passports, but I mean, maybe it's the kind of thing where you have to say, you can only come if you're vaccinated. Because you can't let everybody come.
Well, and the problem with that too is that
but then if people have cancer or if they have autoimmune disorders or anything that keeps them from getting vaccinated, how do you give some people unvaccinated okays to come to events and other people not?
Yeah. Well, and then where are you going to have it? Because also there's a certain governor that has decided that vaccine passports won't be a thing. So then, local government has weird powers related to what is safe. So.
Yeah, I mean, I've been hearing a lot of weird things like what is it? Columbus, Ohio, they're going to do sporting events in outdoor arenas like baseball kind of thing, but have vaccinated sections and unvaccinated sections. And that to me seems weird.
Whoa.
That just seems weird.
That seems extremely weird. That seems like way more risky.
It does seem risky. It's like, okay, so you're going to take all the unvaccinated people and put them all together where they can infect each other. So that's your plan now. How is this going to work out for you? I'm just like, what scientists are you talking to? How's that working?
None is the answer.
Yeah.
None scientists.
Yeah. I'm not skipping summer. I am all about summer.
I'm pretty excited. I've been cold for a long time. I'm excited to not be cold for a little bit.
I mean, I don't like 100-degree temperatures. I don't like humidity. I don't like fires. I hope that we don't have fires like we've had. I mean, this year's huge drought, so the entire West Coast is dry. And that's going to be interesting. Yeah. I don't know, Hot Rod, I feel like this summer with COVID, people who are vaccinated, I hope have a good time. I'm very excited to be able to have outdoor barbecues with people who I haven't hung out with in a year because we're all vaccinated now. I'm not excited about going to a movie theater with a bunch of people I don't know. That's not something I'm interested in doing, but it's summer, so outdoors and enjoying those kind of outdoor experiences with people, I think will be great. Yeah, socializing with other humans. I think that's going to happen more, and I think a lot of it's going to be okay. And a lot of people are saying, we got the vaccine so that we could be with other people. And yeah, the more people get vaccinated, the more people get vaccinated, the more people will be safe congregating together. So yeah, it's just better that way. Good night, Eric. And the Accidental Viking, I see now that you changed your name because Americans could not pronounce Nadelva correctly. So I'm doing it wrong then, huh? I apologize. Sorry about that. Yeah, and if you're outside, I mean, if I'm in my backyard, I'm not wearing a mask, right? I'm outside. If I am need Elva. Thank you. So the CDC telling us to be maskless, we talked about that last week, right, Blair? And
yeah,
I think it's up to everybody still. I don't think we should judge people. I think I hope that the people who are not wearing a mask are vaccinated, when you're outside running around. But at the same time, I'm also, if I'm at a park or walking on the sidewalk, but there's not a lot of people on the sidewalk, I'm not going to stress about that.
This is like the super un-PC way for me to about it. I think it's, I was just like commenting today how now when I go out and I walk my dog, there's this internal question that I always ask when I see someone without a mask on. Or I'm like, are you not wearing a mask because you're vaccinated and you read what the CDC said? Or have you just never worn a mask?
Yeah.
Should I be mad at you for not wearing a mask right now because you've never worn a mask and you don't care about other people? Or are you just following what the CDC says? Because I can't tell.
You don't know. That's the thing. We don't know. And because as a society, we're so mixed right now and we have extremists at either end, of the spectrum of things that they're doing or not doing.
Yeah. I mean, it's tough because, the thing that I keep saying is if I was walking or hiking or running or walking my dog or doing anything outside where I am 10 feet behind somebody else that's walking at the same speed as me,
yeah,
I would want them to be wearing a mask because I'm in their airstream for a long time.
Yeah.
And I think that's the part that gets me, is that it's exactly what we were talking about last week. Like, when you have all these conditions on what is safe, people only hear, okay, I don't have to wear my mask outside anymore.
Yeah.
It's not, if you're vaccinated and you're more than 6 feet away from people and you're only going to be in shared space for less than 2 minutes and then you can not wear a mask.
Yeah, we talked about like all the seven or more mitigation factors that schools do to reduce risk, and it's like, how do you get that? That's the thing that has, I think, been the hardest part of getting people invested in social distancing. So the social distancing as a phrase used in public health includes physical distance. It includes time. Distancing yourself, like, so you're not spending a lot of time with people, and it's masks. It like incorporates all those things, and that's, people don't understand that necessarily, and they're like, well, I stood 6 feet away, and they get that one basic rule in their head, but they don't understand the nuances of why it's 6 feet, and why in a grocery store does it not really matter whether it's 6 feet or 60 feet. Well, it's because all the air in the store is getting mixed, and it depends on the ventilation patterns. And yeah. I don't know. Nidelva, you're saying that you think areas of the U.S. are going to be constantly fighting this virus, and I think areas of the world, areas of the United States, I think that's absolutely true. We're going to have pockets that are vaccinated, pockets that are not. And it's constantly going to be going. Constantly a thing.
There was somebody on NPR today saying that, it's what we've been talking about forever, that you're going to get your annual COVID shot and it's never going away. We could have done that. But
we could have gotten it to go away. But no.
Specifically the way that, one, that vaccination rates are dropping in the United States indicates they don't expect us to get to herd immunity in the United States. But two, these other variants and surges in other places in the world and the continuation of travel means that most likely this planet is not getting to herd immunity. Like, nowhere are they going to get to herd immunity. Maybe Australia, if they continue to lock it down over there.
Right. Australia, New Zealand. We've got a few places that have done it. Yeah. But if
back in my day, we were allowed to travel to Australia. Just fully, like,
Yeah, and it's if we're not going to get to herd immunity, what that means is that it's constantly going to be bubbling up. We're going to find new variants. We're going to need new boosters because it'll mutate, and we'll need something that helps us get around that mutation. It's so sad, but it makes me so sad that because people couldn't wait, because people were not patient, because our government couldn't support people to allow them that space and freedom to be able to stay home and not work, not worry about rent, that what we ended up with is a situation that's even worse than anything. It's going to continue, and we're opening up all over the country anyway, because everybody's like, well, now it's with us. And we're over it, and I
got my vaccine. I want to go do all the things.
Yeah, I got my vaccine, and people are numb to the death numbers. People are numb to the long term. It's, and Gaurav, you're talking about India. There's so much death right now. I'm so mad at people.
Well, it comes down to
the people here right now watching. I know you're good.
I'm not gonna say I'm 100% thrilled with the current leadership that our country has, but I think they've done a heck of a lot better job than the previous. And I can't help but wonder what would have happened if somebody willing to make a tough decision was sitting in that space when this all started. Because that's really what this comes down to, right? Is there's politicians all over on both sides of the aisle. I live in California. What's happening here right now? There's a recall election. Let me just do everything that everyone wants, right?
Totally.
So it's there's a pressure, and politicians are bowing to the pressure and not making the difficult decision. If somebody said, I'm locking down the entire United States, you cannot cross state lines, schools are closed, I'm sending everyone checks to not go to work. Like, if it had all been done like that for 6 weeks, it's the whole thing Justin was talking about the whole time, right? If that difficult decision was made at the beginning, it would have cost less people would have died, and it would be done.
Yeah.
But
there were so many missteps.
Nobody wanted to make the unpopular choice. Nobody wanted to step up and be the kind of the bad cop, the tough parent, right? Who's like, I'm sorry, but I have to ground you. Like, we had to be grounded, and nobody wanted to be.
I'm sorry, United States, you are a petulant child.
Yeah, like nobody wanted to get yelled at that way, and so here we are. It's very frustrating. And of course, everyone's sick of it. Even people that know this is real and know it's bad, there are people who are just over it. And who can blame them? Who can blame them?
I don't blame. Yeah, it's tiring. People want lives back. They want experiences back. They want opportunities.
And we will be studying the neurological effects, metabolic effects, all of the effects of the past 18 months plus when we're all done with this.
Metabolic effects of my basement. Metabolic effects of my basement.
I mean, we all went a little crazy, right? So like,
I am not sane. I just want you to know that.
I've gone through looking back on things. I don't think I ever thought I was depressed. I was very lucky. I did not lose my job. I get along very well with my cohabitant. I adopted a dog months before the pandemic started. I ended up getting a new job. Like, all these things happened that I am extremely lucky about how this pandemic has gone thus far. I have lost one close friend to COVID, which is very, 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 very sad. But I know other people have gone through much more than I have. But thinking back on things, I never would have said I'm depressed, but there was definitely something wrong with my brain and my emotional state this time last year. Something was wrong. Yeah. And looking back on that now, I can see that. And also now, by the way, having been vaccinated and going out to do some things, it's scary. I've been rewired, and I'm like scared of crowded spaces in a way I've never been before. Yeah. I feel weird doing things. I'm an extrovert, and I feel drained now after I hang out with people because I'm so
Welcome to my world.
I just want to go home. And so it totally rewired me. Like, I could have gone out to lunch with a friend, out to dinner with a friend, out to brunch the next day, out to dinner with a friend. And like, I could have done that for a 3-day weekend and not felt tired, felt energized, felt excited. And that's, true extrovert stuff, right?
Yeah.
But now I do one social event and I'm done for the weekend.
That's it. No more people. I did it. That's good.
It's totally rewired my brain. It's crazy.
Yeah.
So, I mean, anyway, my point being, I totally get why people are totally sick of this and need change and have to get out. And it's been too long. It's been too long. It didn't have to be this long.
No, it didn't.
I get it.
It is. It didn't. Yeah. Yeah. And we're all frustrated about that and tired. But I'm looking forward to summer outdoors. Maybe, just maybe.
Mm-hmm.
There's some summertime to be had. I get to visit my dad again.
Yeah.
Some good things. Good things are coming. Although travel does make me stressed out. So, being at home this last year was great. Pam in the chat was saying she thinks it's great where we've been at home to be able to spend more time with each other at home, which that was one thing that was wonderful this year.
Mm-hmm.
But yeah.
I do think I've grown
balance.
I've grown weirdly codependent with my partner now. In a way that's like, we get along so well, which I'm not going to complain about at all. But now we are speaking inside jokes to each other all the time. Weird voices.
Complete each other's sentences.
Yeah, it's now I'm going into work like a day a week now. And so now when I go to work, I'll just see something and be like, oh, I can't say that here. Or like, oh, no one will know what I'm talking about if I bring that up. And it's very funny to have been around him and my dog. And that's it for over a year.
Yeah.
Pretty wild.
I think I'm codependent with my basement.
Yeah. Well.
Oh, basement, you're so nice to me. You give me a place to be. And a place for my computers. Identity 4, that's so sad about your mom not wanting to get vaccinated. It's not, just, come on, family members. Everybody get vaccinated so we don't have to ban each other. Come on. Come on. Yeah, I like working from home, but I've always worked from home. I wanted to make life from home, but now I want other people to go away.
I was pretty excited to start going into the office again, just because also I'm just in a different room, which is really cool. And I'm not surrounded by, I don't know, things that need cleaning and just, other things. I can kind of focus. But I am hoping that, like a lot of jobs, I'm hoping that my job will be able to be hybrid after this, because it's been proven that I can get a lot done at home and use my own Wi-Fi and my own power. And it's because I work for the government now. Fossil fuels are something that they have to report. So if people can do telecommute days, then it reduces commute hours, and so that can help them with their quota for that. So I am hoping that I get to do some telecommute days every week after all this is over. I think it's proven that a lot of jobs where people thought like you have to be in the office, productivity, you have to be in the office.
Yep.
A bunch of jobs are like, oh, I guess you could do a day or two a week at home and it would be fine.
You were fine at home. Oh my,
Yeah.
we don't control your
you're
whole
still
life.
productive. Despite your significant others and your children and your pets, you were still productive. Huh.
I think there's that. It's an idea of. There are some managers and companies want to, it's not just a job. This is now your life. You know,
Mm-hmm.
it's not. It's they own you now. You got a job, but they own you and your time while you're working, and yeah, it's hard to do that when you're at home.
I found myself ranting a lot lately, and I probably ranted on an after show not too long ago about this, but I was ranting recently about how automation and computers and the internet and Excel spreadsheets and all of these things that we didn't have 20 years ago even. I mean, we had some of those things 20 years ago, but not to the level that we currently have them, right? And that has increased productivity for any job that involves a computer so much. And how it should have worked is, oh, you can get an 8-hour day done in 4 hours? Great, you work a 4-hour day now.
Work 4 hours. Yeah, no.
But no. Oh, you get 8 hours of work done in 4 hours? Now do 16 hours of work in 8 hours. Who decided this? I know who decided this, but what? How did we accept that?
How did we accept it as workers?
Okay.
Because of union busting. But did I say that? Okay.
Union busting, and also, companies taking advantage of people wanting to be good at what they do.
There is
right.
that. Yeah.
So
if you're passionate about what you do, you're like, I will work. I want to do this.
Yeah,
I want to
I'll
make
do
this
more.
project.
I'll do more.
Yeah.
Oh, I got my project done. What can I do next? Which is great. But also, if you used to have to input data off a piece of paper and it would take a full day to do a stack of data, and now the data automatically gets put in a spreadsheet and into a database, and it takes 5 minutes.
What do I do now?
That doesn't mean that you expand your job description and work that much more. It's very frustrating to me, because the amazing technological advances we have made should mean that we have more time to be with our families.
Mm-hmm. That's what they should have done for us. But instead, we bring these devices home that connect us to our jobs. We, never mind the pandemic, but there's all the things we don't know how to turn off. People take emails on the weekends. I'm like, hey, no. Mm-mm.
Mm-hmm. Mm-hmm.
Yeah, I stopped doing work on the weekends, and then I started working with academics again, and they started emailing me on the weekends, and I'm like, who are you? Yes. Go away.
Yeah.
I'm not working on a weekend unless I have to. But yeah. Oh my goodness.
Yeah, I don't understand any of it. I don't get it. I don't understand how we as a society just went, okay,
I'll do that.
More work for less money? You got it.
No, I want to have my time.
Just the fact, I just got so depressed thinking about how, a third of my day is spent sleeping, and another third of my day is spent working.
Mm-hmm.
That's not. And then how much of your day is spent getting ready for work?
Yes, Gaurav.
How many hours do I spend driving to and from work, or making lunch for my work, or packing my bag for it all adds up. And then that's
congratulations on retiring, Wiz Mike. Someday.
Oh man, that's one of my favorite things about my new job is the retirement plan.
That's great. I just feel like I'm like, okay, when I retire, what projects am I going to do? I'm like, ah.
Well, I'm already really excited because, I'm
other things am I gonna
I'm running
do?
volunteer programs. I've learned all about what it takes to be a campsite host.
Yeah.
And I just think it would be so fun once I retire to buy a trailer and just
be a camp host.
Yes. Do like 6 months here, a year there, and just do all the national and state parks and like just hopscotch all over the US and Canada.
That would be fun.
It'd be a camp host. It'd be so fun.
Mm-hmm. Welcome, Donald Brandt. We are in our after show right now. We're just talking about dreams. Dreams versus reality. Camp host would be awesome. That would be very fun.
Ugh, so cool.
Yeah.
Because then it like gives you something to do.
Go big.
So you're volunteering maybe, for us, it's like 20 hours a week. But yeah, you volunteer to help out in the park and then you get to live there for free. Yeah, you get to welcome people into the campsites, sell firewood. Just, oh my gosh, I think it'd be so fun.
It might get old quickly, like Shubru's saying in the chat, but I don't know. Could be awesome.
Depends. It depends on the site. It depends on the type of person you are. Like, I like to stay busy. And, we have people in the parks that I'm in charge of now that have been camp hosts for like 6 years in the same site.
In the same park.
Yeah, we had to put a limit on them now. Now there's a 3-year limit, but some people
get to live in the camp.
But yeah, there's some people who want to do it forever.
I have not seen Nomadland, Nidelva. I heard it's a little depressing, so I decided not to watch it. I don't like depressing things. Yeah, I don't like that. I've heard it's very good though, but I don't know if it's one for me. I like things like what's the new one on Netflix? Whatever versus the machines. It's really good. It's really good. Also, Shadow and Bone on Netflix.
Oh, I've heard of that.
Really good. Fantasy. It's super fun.
I saw the trailer and was very confused about what it was about.
Well, it's a young adult fantasy series. Mm-hmm. They turned into TV, and it's good. I really like it. It's really good. Yeah. We want better than what we've got, Brian Burwell. Yeah. You can get there. You can get there. Hey, Blair. Is it time for bed?
Mm-hmm.
Should we go and put our anterior insulas out?
I mean, mine's been.
I really haven't been conscious for this entire conversation.
No. Blair's not here right now.
Blair's not here. Please take a message.
Oh my God.
Hopefully there will be new t-shirts in the store soon.
Mm-hmm.
Maybe it just needs more time.
I think so, because I can't find it anywhere. It's like it's in a purgatory. I think it'll pop up.
Purgatory.
But the mug popped up. So that's progress.
It's progress. We shall get there with science-y goodness for all. Full of science-y goodness. Mm-hmm. Say goodnight, Blair.
Good night, Blair. Say goodnight, Kiki.
Good night, Kiki and everyone. Good night, everyone. Good night, everyone. Thank you for joining us for another episode of TWIS and our after show discussion. Yes. We'll be back next week. I hope everyone had a wonderful Cinco de Mayo. Cinco de Mayo! I had 0 salsa today. What?
Oh, that's okay. It's like not a real holiday anywhere, right? Isn't that a thing? It's not even celebrated in Mexico.
Yeah, because it's an American holiday meant to celebrate the relationship between the Mexicans and the Americans. But yeah.
Right. Most people just use it as an excuse to
get drunk,
drink margaritas till they barf up salsa. So,
and I don't like tequila, so there you go. News for everyone. As we say goodnight on this Cinco de Mayo. Have a wonderful week. We will see you next week with more science. I hope you stay well. Thanks again.